Hey guys, today we're going to learn about forensic entomology, and it's pretty much the study of how we use insects to solve crimes. It doesn't necessarily mean it's a murder, so I want to take a look at all the different types in this next slide here. So, anytime we use forensics involving any kind of case or something to do with law is forensic entomology. Some things that you may not think about. So, this picture on the bottom left is actually the mosquito that was involved in the Zika virus. Do you remember that a while back when we had that? Um, that was a law. The law was involved with that because we were trying to eradicate that mosquito prior to it coming to the United States, but we weren't allowed to because of the federal laws on what are they called? The stuff that kills mosquitoes, which I can't think of the name right now. Um, you know what I'm talking about, pesticides. There we go. Okay, so the laws on pesticides wouldn't allow us to just dump those pesticides on United States property. Now, in other countries, they also knew about Zika and were able to prepare prior to that and did eradicate that species of mosquito. So very interesting of how that can come into play. How, you know, insects can have to do with the law. So the top picture there, well, the middle picture, let's look at the middle picture. The middle picture on the bottom, that's a picture of termites. And they also come into play in the legal aspect, especially when you're buying a new home. So you may not know this, but when you buy a new home, you have an inspection done. And if your inspection comes back with termites, then the house that you're buying, the people will probably have to fix it or do something. Or let's say you buy a house and it had an inspection and you find out later you have termites. Well, that's obviously a problem. So that's where the legal aspect comes in for the termites. Another thing that is gross that can happen is this picture on the right here. This is a picture of a banana. And those are fruit flies, which are really common to be on a banana. But the story I have to tell you comes from a forensic entomologist friend that I had who is from UF. And he said that he had this case one time where there was a woman who found bugs in her cheese. So she got home. She went to, let's say she went to the grocery store. She put her cheese in the fridge. She took the cheese out to use it maybe the next day. And when she unwrapped it, there were bugs inside. So the law part of it came in where she wanted to sue the grocery store because when she tried to take the cheese back to the grocery store, they said, no, 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 we won't take the cheese back because we don't know if the insects were actually from your house. So where does a forensic entomologist come into play here? Well, they can look at the species of insect, first of all. Is that native to where the person lives? They can look at the age of the insect, right, or the larva that's there, and figure out when was that insect on the cheese, because if it was on there prior to the purchase, then it probably did happen in the store or in the shipping facility. Or did it happen after when the woman had it? So that was actually a legal case. Sadly, I don't remember the outcome of it, but I thought that was really interesting that literally bugs and food can come into play. Something that's going to gross you out, and I'm sorry, but I have to tell every class that I teach this. There's bugs in a lot of the food we eat. You just don't know it. So, for example, a lot of us will get grossed out by this, but if you eat cereal, in cereal, oftentimes there's insect eggs that don't ever hatch, but a lot of times there's insect eggs in the bags from the manufacturer plant. If you think about it, we can't manufacture things in these big facilities without having issues with any kind of insects, because we can't use a lot of pesticides in there because that's not going to be harmful. That's going to be harmful to us. So that's just something to remember. Another thing, and I think you guys, if you were in my chemistry class, we talked about like a bar of chocolate has at least 7 insects in it. Because again, you can't keep all the bugs out of these processing plants. Okay. So I just wanted to talk to you about the ways that insects are used in the law, aside from death investigation. So we will be looking at, from here on out, the death investigation part of it, but I just wanted to let you know there are other parts that are involved in forensic entomology. So going back to the history of forensic entomology, so the first documented account where they used forensic entomology was by Sun Tzu in China, and it was 1235 AD. And let me tell you the story. So there was a story where there was a farmer who was killed while he was working in his field. They found him laying face down in the field, and he had a sickle by him. That's what he would use to harvest the wheat. And the judge in the town wanted to catch who did the killing. So he was smart. He said, it was a small little town. He said, I want everybody that is a farmer in the town to bring your sickle to a town hall meeting. All right, so they all brought their sickle, which is that picture that's showing them lined up here. And the judge watched, he had them set them down, and he walked along the sickles, okay? And he was looking at them. Now, they all looked pretty clean. There was no evidence of blood on them. However, insects can smell any microscopic bit of flesh or blood that would be on any type of tool like that. So even though it looked clean, it was not, according to the insects. So they were easily able to tell who did the crime because the flies landed on that sickle, and the judge came over to that person, and right when he accused him, that person confessed because they knew there was no way getting out of it because the insects had already told the truth. So I love that story because it's just interesting how we used it long ago, and it wasn't a science yet, but that judge knew that the insects would be attracted to the blood. So some other history dealing with forensic entomology. One of my favorites here on the left is Francisco Redi. You may have heard of him in biology. One of the experiments he did, so believe it or not, you may think this is shocking. People used to think that maggots spontaneously generated. Like, you know how you might go out at night and you look in your garbage can outside and you might see maggots? Well, that's kind of like what happened to them. They would not see any maggots in a place, and then they'd go back, a day later, and there would be maggots there. So they thought it was a spontaneous generation of maggots. Now, to us now, that sounds silly. So I want to talk to you about the experiment he did to disprove that. So what he did was, you can see these 3 jars here in the picture. He put some meat inside the jar, and we're going to do a similar experiment like this. You're going to make your own body farm, but I'll tell you about that later. So here are the 3 different canisters, and one has nothing, no lid or anything on it, and it has the meat inside. One is completely sealed with a piece of cork, so nothing can get in or out. And the other one has a covering over it, but it has holes in it. So think like cheesecloth, or it says here a gauze, anything like that would have holes in it. So in all of these situations where the insects can smell the meat, they're going to be attracted to it, right? In the middle flask, they shouldn't be able to smell the meat at all because it's literally sealed, right? It's kind of like no air can get in or out. So we don't have any insects coming near that. You might have something like a dog or something like that because they can see the meat, but an insect, they're attracted by smell. So in the first flask, it's open, so they're able to land, and you can see in the picture that there's some maggots already deposited on the meat, right? In the picture on the right, you don't see any maggots on the meat because remember, the insects can't fit through this cheesecloth, but they smell the meat and they know that's a good place to put their maggots because it will be a source of food for them, right? So they lay the maggot eggs here, hoping that the maggots can get through. P.S. Lots of them can, because the maggots usually can move around depending on the size of the holes. These guys will move around and make themselves be able to drop through the gauze, and that's gross, but that can happen. Okay. So anyways, he was able to disprove that it wasn't spontaneous generation, that it was literally the flies lay the eggs. And because it's such a short time, we don't always think that there were ever eggs there if you never saw them, right? Because the maggots at first aren't moving. They're just the pupae. And then once they actually start to move around, then you can tell it's, something that might have been. But again, once it moves around, it doesn't look like an egg. So people didn't believe that something laid an egg there. Okay, Linnaeus is the person that is responsible for coming up with all those. Do you remember those charts you looked at in biology where you had to figure out the species and the phylum and all that business? Linnaeus was in charge of making lots of those, specifically lots of different insects. So I know it's for all animals, but he did a lot of the insect stuff. Another one of the interesting cases I love for history of forensic entomology is about Bergeret. So Bergeret was actually a doctor in Paris. And there was a specific case where there was a couple who bought a new house, and per usual, the woman wanted some modifications done to the house before they lived in it, right? So they bought this house, and she wanted the fireplace to be redone. See, people did this back in the day too. They still wanted to redo stuff. So when they started to redo the fireplace, the workers were taking parts of it off, and they were able to find inside the fireplace there was a wrapped up child, a mummified child, really small child. So what happened? Of course, the people that were working there reported it, and they came to the people that bought the house, and they said, "Hey, we're going to accuse you guys of murder." And they're like, "Whoa, whoa, whoa, hold on a second. We just bought this house. We didn't put this person and there's baby in here. So how could they prove that?" Well, that's where Bergeret comes in. So somehow, I'm not sure how you got contacted, but Bergeret, that is the doctor, dabbled in entomology a little bit, and he was able to look at the insects that were on the child and able to find that the child had at least been in there a year. And these people had just purchased this house, so there's no way they could have murdered the child. Very super interesting to me that they were able to solve that in the 1800s like that. The last one we're looking at is Meining. And Meining, he is known for looking at a carcass of something that's dead and paying attention to what insects arrive at what times. So, for example, we know that on a human body, the first thing to arrive, depending on where you live, pretty much everywhere though, is blowflies. Blowflies are going to arrive first, and then after that, there's going to be other insects. Where, like at the end of that, is going to be like beetles and spiders. But we know that there's a specific, they call it a succession, a specific order that the insects arrive in. Okay, so that's what he was responsible for. All right, so we're going to look at the different stages of decay. And when he talked about succession, it'd be like right after the animal dies, what would we see, right? That would be the blowflies. In the bloated stage, and this is where we start to see the gases building up. That's why they call it bloated. And you can just look at all these stages and you can look at the actual insect activity. You can see some maggots on here, not much. And you can see a lot more activity on these later ones, except to when you get to the dry stage. Dry stage, again, is when you're only going to see like beetles and maybe spiders and maybe wasps or something like that. They're curious. Okay, so here is one of the very original stages. So you're going to see more eggs and stuff by any openings on the body, any orifices. So, ears, nostrils, the anal cavity, maybe a belly button, if the animal has it. So take a look at this. You can see that these things right here look like sawdust. Those are eggs, and so are these. So I think this is by the pig's ear. You can see that there. So autolysis is when the cells start to break down and die, and when that happens, we get a gas buildup, right? There's a chemical reactions taking place, and gases build up, and that's what leads us into the bloat stage, which is the next one. You can see now that we have not just little maggots, we have other flies present. There are some still maggots here that are not just eggs, they're now maggots, they're larger, and there's different type of flies that are here. The big thing here is putrefaction begins, and that's where, if you've ever seen a body that's dead, maybe on those body farm videos where the body looks yellowish, that's putrefaction. It's the fats, they like rise to the surface and they turn a yellow color, the fats that are in the body. And remember, these gases, so the body is swollen because of the gases, and those are what draw the insects to the body. At some point, you could go up and pop it and it would let all the gases out, but at some time they will slowly release and the gases will go away on their own. It's a terrible smell though. So the decay stage, there's a lot less flesh there. Still there, but the maggots will eat the flesh, and this is where you might start getting more of a maggot mass. There's lots of maggots, and this is where the temperature may go up as well. We're not having gases emitted anymore though, so the blowflies aren't attracted. It's pretty much just like different insects that are just kind of around, not just attracted to it, but they like happen upon the cadaver. And here's the post decay. So this to me, if you were a forensic entomologist and you had to arrive and take a look at this would be the least gross to look at. It's not going to smell as bad, right? It doesn't have the flesh on it. It looks slimy or anything like that. You're really mostly looking at bones here and maybe a little bit of skin and fur, depending on what the mammal is. What is yucky is that these guys are a lot bigger. These pupae are going to be a lot larger. And what happens at this point is they start to bury themselves in the ground next to the carcass. That's always gross. They get a hard outer shell. And this is just the bones that are left. And again, at this point, you would see beetles and maybe spiders and moths, it says here. Okay, so several things affect the rate of decay. Biggest is temperature. I know Dr. Bill Bass talked about that. And the reason temperature is a huge factor is because all these insects' life cycles are based on temperature. So the colder it is, the slower they're gonna be attracted to the body, number one, which starts the decomp process, and also the slower that they're going to go through their cycle. Okay. If a body can't be accessed by insects, let's say it's like this picture here, it's covered up, that limits how much the insects can smell, so they can't be attracted to it. If you then put the body in the trunk, you really can't smell the blood either. So the other things that will affect it is can other insects actually, or other animals actually smell the body? Because if they have access to it, they will eat it and help the decay process go quicker. Was the person sick before? Because if you have a high fever, that's going to help increase your process of decay at the beginning anyways. Lots of different things. So trauma, if you have a head wound and the insects can access that area quicker, that will speed up the rate of decay. So I could talk about that one forever. There's lots of different things to talk about for this. Just want to talk about a couple of them. Okay, we want to be able to estimate when did the person die. Postmortem interval is the time period since death, right? Has it been 6 hours, 18 hours, 2 hours? Typically, they won't say just an hour. They'll say a range. You always want to give a range of time. It's been between 0 and 2 hours that this person died, right? Or it's between 18 and 24 hours. You're never really going to give an exact time. So when they do that in the TV shows, it's not super accurate. The way you do this is by looking at, okay, so where is the body? Did the person die in a desert? Did they die in a freezer? What stage of decomp are they in? Are they in the bloat stage? Are they in the fresh stage? Are they frozen and they haven't started decomp yet? Really? Because the body's been frozen and hasn't been able to do that. For the forensic entomology side, what insects do we have there and what larvae and how much? All of these things are going to be big factors. What's been the average temperature over a given number of days since this person's been missing? That's another way we'll figure it out. And so it'll be a calculation we'll look at for this. Like I said before, these insects are temperature dependent as far as their development. So you can look at here's the life cycle, right? They go through these different life cycles to the eggs and the larvae and the different, we call them like instars. Those are the different levels, different stages of larvae or that a maggot can go through. And depending on the species, each stage will take a different amount of time. Okay. There's something called a threshold temperature, which you can see a video link in. But the key is at a certain temperature, they don't develop anymore. So like if it gets too cold, the insect can't develop. You'll notice here, for example, insects aren't usually flying below the temperature of 60 degrees. So once it gets in the 60s here, around 60, 65, something like that, we won't really have mosquitoes flying around, which takes forever to happen here, but that's typically the temperature. They also develop quicker when it's hotter and slower / non-existent development when it's cooler. Okay, so here are some beetles and I just want to show you some different pictures of things. These are some maggots here, and even this yellow business here, those are different maggots. Those look like green bottle flies, and we'll look at some pictures of those in a minute. Just some interesting facts about the blowflies and the first arrivals. So it's crazy that literally each blowfly that lands on the body can lay to between 200 and 250 eggs. That's insane to me. Okay, that's a lot of eggs. they hatch within the first 24 hours. Assuming now that first thing, as far as arriving to the cadaver in minutes, we're assuming it's not raining. Now, rain is one of the things that will delay insects being able to be attracted to the body. Because number one, the insects don't fly in the rain. I don't know if you ever noticed that. We don't really have flies around you or bothering you when it's raining out, right? So they don't fly when it's raining, and they are less active at night. So that is something to keep into account. This is, I think, interesting, the maggot mass. When there's lots of maggots on a body, it will raise the temperature of the body, which will increase decomp. And here's another picture of the life cycle you can see. At this point, when they reach this pupa stage, this is when they oftentimes burrow under the ground underneath the body before they develop into a fly. And here's just some life cycle. How long does it take to go from each stage? So one of the things they do when they're looking at a body and they have to collect a lot of these maggots, they put them in what's called a killing jar. This is a mason jar here. Typically it's full of some type of alcohol. And what it does is it stops their development at a certain time period. So they don't develop any longer. That's the key to putting them in that kill jar, is that we need to stop them at a certain point, right? We don't want them developing further, because if they do, then you can't tell what it was when you got it, what age they were when you got it. And this is just talking about how you would do that. You are going to capture your own insects in our lab that we do. And here's a picture that I love that kind of shows all the different tools and things that you'd be looking at when you're going to be doing that. Okay, so something interesting you may not have thought of. Maggots, they feed on the flesh. So if we can get, assuming that the corpse is fresh enough that they actually fed on the flesh, we could analyze a maggot for several things. We can look to see if somebody was using drugs at the time based on the flesh the maggots have eaten, and even what drug it was. Was this an overdose? Was this a poisoning? Right? So very interesting that we can tell that. Another thing that they're doing better and better on now is from the flesh inside the maggots, it's now enough flesh to be able to figure out the DNA, which is crazy. To be able to let's say we don't know who the person is, and we can look at what the maggot has eaten to determine who it is. Okay. There's a couple case studies here. I'm not going to read these to you. You can read this on your own time if you want. I just think they're interesting, talking about how they actually caught the person with the insects, forensic entomology. Okay, so what we want to be able to do is, what I want you to know is that from insects, we can figure out the postmortem interval. We can figure out if a body's been moved or not. Now, I didn't really talk about that, but let me talk about it for a second. So, for example, let's say that on a body, there was a certain type of, because you're going to look at a case with this, certain type of grasshopper. But that grasshopper isn't indigenous to Florida. So that means that body was probably moved, right, at some point. So those are the type of things that we can get from information, especially, too, if the killer says, oh, I was in the state of Florida the whole time. That's not possible with this type of insects on there. Okay. And then, like I said, possible poisoning or drug use from insects and what they eat. Okay, so what you guys need to do after this is you need to work through the Module 5 study questions. You can see I was going to add something else on there, but that's what you need to be working on. And the next thing we're going to look at is setting up our lab. You're going to have a lab that you set up at home, and we're going to grow our own maggots, okay? And you're all going to have different conditions. You're going to take pictures. This lab is going to be a class effort, but you're all going to have your own sample, and you're going to have to take pictures of it. So there's no getting pictures from anybody to be able to use their work, because you have to have your own cup. I'll tell you more about that later. All right. Hope you guys have a great weekend. Bye.